So without further ado, I will introduce our first speaker, who's Dr. Yvette de Haas, who's a Program Manager in Animal Breeding and Genomics at the Wageningen University Research in the Netherlands. Her work focuses on improving the sustainability of livestock systems through advanced genetic selection, including strategies to lower methane emissions while preserving animal health, productivity and resilience. Yvette leads international research collaborations and contributes to efforts shaping breeding programs that integrate environmental goals. Thank you, Yvette.
Thank you for the kind introduction. Let's see if I get this one working. The other way. Yeah, the other way. I would like to talk a little bit about genetics and breeding as a mitigation strategy. I am involved in the Global Methane Genetics Initiative, together with Rob and Hayden and Andy, but also my colleagues back home, Roel and Birgit. When thinking about methane, there are a few breeding strategies, mitigation strategies that we will hear about today as well, about the ration composition, feed additives, and also vaccination or vaccines. But what I would like to talk about more is about breeding. What we do with breeding is that we improve the next generation, or the genetic level of the population of animals. And we do that by selecting the best animals as being a parent for the next generation. And in order to be able to select these best animals, we have to know that there is variation between animals. What you can see here on the lower graph is like just a small example of a small data set where all the animals are ranked from low to high emission, and you can see that there's quite a variation, and therefore we will also be able to select like the best, in this case like the lower emitting animals, in order to be a parent for the next generation. And this leads to a stepwise and cumulative genetic progress. So even though the steps might seem small, they can be added from year to year to year, and therefore also add a lot of improvements. And we have been doing this for many years already within animal breeding, starting with like production traits in the early 80s. But then in the year 2000, or around 2000, genomic prediction made his entrance, and that did open opportunities to start selecting for other traits that will not necessarily have to be selected on or collected on all the animals. And therefore, since then, we also moved towards traits like feed efficiency or methane emissions. My background is in dairy cattle, so all the pictures that you will see relate to dairy cattle. In a nutshell, genomic selection is that we have a data set which is still big if you compare this for like nutritional studies, but it's small compared to like a whole national herd or whole national population, where we know all the phenotypes and we know all the genotypes. So we know like what is the genetic basis of the animals, but we also know the important and traits that we are interested in, like methane today, but can also be resilience or feed efficiency or health traits. And then based on this reference population, we create or we work on a prediction equation, and which is also called a genomic breeding value. And then next to this reference population, we have like all the selection candidates, which are all the animals in the country. And they might have, from those we know the genotypes, but we do not necessarily know the phenotypes. But with this prediction equation, we will be able to select, to make a selection of the best animals, so like the right side of the normal distribution tail, and therefore be able to select for, in our case today, the green cattle for using the genomic breeding values. So even in the reference, so even though it does release the number of records that are needed, in the reference population we still need information on methane emissions, and for that there are several ways to record methane. And there are only a few that can really actually record methane production in grams per day, which are like the respiration chamber and the portable accumulation chamber, but also the green feed. Sorry. But none of these record for 24/7, but so some of them also like the PEG and the green feed, they are like spot samples. But also, if they do record for 24/7, like the respiration chamber, they don't record it for like a very long period, like a full lactation, or let's say even 3 months. So none of them, all of them are kind of predictors because none of them measure like the full potential. But there are also other predictors where we measure the methane concentration rather than the methane production. There's those equipments like the SF6 or the leasing methane detector or the sniffers, which are worldwide used a lot. They are way less cheaper than the ones on the left top. They are also they are less labor intensive. So there's a benefit there, but know that they do measure the concentrations and not so much the green feed production or the methane production, but can for sure be used as a predictor of the methane production. There are also other predictors which are more and more developed and more exposed, which is the MIR predictor of the fatty acids in the milk, but also the rumen population composition and all the microbiome composition. So there are several ways to measure, and there are several ways to get information about the methane production of cows. Well, what can breeding then contribute? This is the thesis of Anouk van Breukelen. She defended her thesis in Wageningen last year, October. And she, what can breeding contribute was really like the main question that she addressed in her thesis. And what you see here is a graph where like the dotted line on top, that's like the current trend. So if we don't do anything with animal breeding, the methane production in grams per day, so not the intensity, but the production, will steadily increase. And that's an increase of, let's say, 13% over the course of 30 years. This is mostly also because more animals produce more, so the intensity might drop, but because they produce more, they eat more, and therefore also emit more methane. However, if we have animal breeding in a selection index, we will be able to bend this curve from even an increasing to like a decreasing trend, where we can decrease up to like almost 30% in 30 years, which is more or less 1% per year. As I said, steps might be small, but by adding them year after year after year, you can really make a big improvement with animal breeding. This is currently implemented in some countries worldwide. Some indirect selection has been done, is already done for like Ireland and also Australia. And like the direct selection or like breeding values for methane based on as grams per day. That's, well, in Canada and Spain, they are already published. And in the Netherlands, we published them earlier this year. And also Denmark, Norway, and France will follow, and possibly others that I'm not aware of as of now. And with a direct selection, and it has been mentioned earlier this morning, we have to be sure that we balance the traits right. So we should not only go for low methane emissions, and therefore not improving efficiency, or not losing production or health or fitness traits. So it's a balancing act, but we are doing a good job, so we are making breeding available as a mitigation strategy. However, this is only done in, like, as you can see, in like the Western or the developed countries, and also only on Holstein cattle. And that's where the Global Methane Genetics Initiative comes up, which has the aim to accelerate genetic progress to reduce methane in ruminants. It's led by Roel and by Birgit. And this one has really the aim to get towards data also collected on other species, on other parts of the world, and therefore be able to have like to accelerate breeding as a mitigation strategy across the world. We have several packages or programs, as you might mention. We have one on dairy, which has, of course, Holsteins, but also other breeds. We have 2 programs in beef. We have sheep, and then we also have like national or region-specific programs on Africa and South America. We are working on a program towards Asia, and then also the microbiome, as I mentioned earlier. That's where the methane is formed, and we will hear more about that later today. So it's a huge consortium with 25 countries involved, 50 partners, and 25 breeds. And this will really make a huge step in towards accelerating breeding as a mitigation strategy. And well, these are all the partners, and with that, I would like to thank you for your attention. Give the word back to Rob.